Hello to everybody at the Stickler Involved People meeting. I'm really sorry I couldn't be there this year. It's really my favorite meeting of the year. I really enjoy meeting all of you and sharing with you what's been going on at the NIH and hearing what's been going on with you also. I am really sad that I'm not there, but at least I was able to make this video presentation and I hope you'll enjoy it. This study was actually started at the National Human Genome Research Institute way back in the 1990s by Dr. Franco Mano, who is my mentor. And I do want to say that we do still have the samples and data that were collected at that time. Any of you that have participated can feel good that some of the information we're discovering is due to your participation. So I'll give you a little bit of a background on Stickler syndrome and then tell you some about the research project. Stickler syndrome is what we call an autosomal dominant hereditary disorder of connective tissue. Autosomal dominant means that you can inherit it from either your mom or your dad, so it doesn't reside on one of the sex chromosomes, and you only need one copy of the mutated gene to have the disorder. So if a parent has the disorder, the chance of inheritance per child is 50%. So it's thought that 1 in 10,000 individuals are affected, but I think that the actual incidence is much higher. As you well know, the syndrome can affect your eyes, ears, facial structure, bones, and joints. We are studying both the clinical manifestations and the genetic aspects at the NIH. I do want to mention that the diagnosis is often missed. The syndrome is vastly under-recognized by physicians out in the community. About 10% of all the babies who were born with a what was thought to be an isolated cleft palate or with Pierre-Robin sequence, where there is a very small chin and a cleft palate, often leading to feeding difficulties, were found to have Stickler syndrome in a study done almost 20 years ago. So the actual incidence is thought to be much higher than the 1 in 10,000 that is often cited. So mutations that lead to Stickler syndrome include those that are found in some of the collagen genes, and you will hear COL2A1, COL11A1, and COL11A2 genes mentioned. There has been also a description of a family with COL9A1 mutation. That family had very short stature as well, and it was recessively inherited. So I'm not sure how applicable that is to most of you. The genes you hear that start with the word col refer to the collagen genes. And at the NIH study in our cohort, about 75% of our patients have a mutation in one of the COL genes, COL11A1, COL2A1, COL11A2, and about 25% we have not found a mutation yet. We are looking at other candidate genes. So here is the structure of collagen I keep talking about. Collagens are actually the most abundant proteins in your body, and there are 26 or 27 different forms of them. The fibrillar collagens all share the same structure. They're made of 3 different strands that wrap around each other in a helical fashion. It's a left-handed helix, and the helix needs to be very tight and organized. It has a very regular amino acid sequence, which are the building blocks. So the building blocks being regular allows it to turn in a very compact and tight fashion. So the collagen molecule, once it's made, undergoes a lot of changes, and its ends are clipped and it gets exported out of the cell, and it gets linked together to form what we call the fibrils, which get laid out between your cells. If there is anything wrong with the collagen molecule, the way your cells glue together doesn't work right, and then there is a weakness in the eyes or the joints or the discs between your vertebral bodies that cause the manifestations of Stickler syndrome. So here is a picture of the collagen fibrils that are actually laid out between your cells. This is by electron microscopy, and you can see how it's like a basket weave, and if there is not enough of it, or if the structure is distorted, it does confer weakness to the tissues. So type 2 collagen, which is the most common mutated collagen in Stickler syndrome, is what's called a homotrimer. It's made of 3 Col2A1 products, whereas collagen 11 is made of 3 different products: Col2A1, Col11A1, and Col11A2. So each of these wrap around each other. So as I mentioned, the location of type 2 and 11 collagens correspond to where you see the manifestations of Stickler syndrome. When Stickler syndrome was initially described by Dr. Stickler, he called it hereditary arthrophthalmopathy, meaning that it was inherited, it affected your bones and joints and caused arthritis, and it also affected your eyes, as many of you know. So manifestations of Stickler syndrome match where you find collagen 2 and 11 in your body, such as in the eye. In the eye, Stickler syndrome can cause nearsightedness. It can cause the liquid inside in your eye called the vitreous to degenerate. As you know, it can cause retinal detachments and degeneration, and also premature cataracts and glaucoma. That is the increased pressure in the eye, and also can threaten your sight. Another finding is the clefting of the palate, or a bifid uvula. The uvula is the small piece of flesh that hangs in the back of the palate, and although the palate might be intact, the uvula may be split in two. We see the typical features of Stickler syndrome on the face includes a rather flat face and a small chin. We also find that the facial features are more prominent in the younger child, but they get reduced with age, and the face becomes less flat and the chin tends to grow. Another typical manifestation is hearing loss at a young age or at birth. This is not uniform, but it is fairly common. So we also find that most of our patients are either tall, statured, or normal, but we have found some exceptions to that, and I'll mention them later. Again, another common finding is laxity of joints, and on occasion dislocations of joints, and there are also abnormalities in the shapes of the bones. So looking at 28 individuals from 28 distinct families, so one representative per family, we almost uniformly found facial features including the flat face and the broad nasal bridge in every person we looked at. An open cleft or a hidden cleft or the bifid uvula was found in a lot of the participants as well. Of course, the core finding was either vitreous change or retinal change. So if you looked at either, all of the 28 persons had that. The high frequency hearing loss was a little less common, 17 out of 28. I do want to mention femoral head failure, which is the deterioration of the long bone that fits into your hip, and that can cause a lot of complications, gait disturbance, and hip pain. Of course, arthritis was very common in our population.